Good afternoon, everyone. Thank you so much for being here. I'm Bob McCarthy. I'm Dean of the UNE College of Pharmacy, and welcome you to this really very special lecture. When I arrived 15 months ago at UNE, I had a chance to meet Ada and Don, and I thought, holy mackerel, how lucky are we to have these two? And the more I got to know them and got to see the work that they've done, I feel even more fortunate. To me, they're sort of modern day Marie and Pierre Curie. And frankly, I'm wondering why they haven't won a Nobel Prize, but anyway, there's still time. They're still young. So let me tell you a little bit about them. I'm going to actually, there was an article that was published in Science about 9 years ago, talking a little bit about them, and I'm going to read some pieces from it as we get started, just because I think it tells you a lot about them. First of all, when you get an email from them, and I'm a little slow, so it took me a little while to figure out that they signed it D lowercase n-a. So DNA. It took me a little while to figure it was Don and Ada. So that's sort of their signature. Most other people figured that out. And the Olinses are best known for the discovery of the nucleosome, which is chromatin's basic structural unit, in the early 1970s. These folks have been at this for 5 decades as a team. They're at a point in their career that a lot of people would say, oh, we'll retire, we're going to go out on the boat, we're going to relax. Not these two. To them, their personal relationship and their scientific relationship really continues. They are remarkable people. So let me read a little bit from this article. So in 1959, Don Olins was a first-year medical student at Albert Einstein Medical College in New York City, and he spent the summer as an intern in one of the college's many labs. Impressed with his work, the distinguished biologist Paul Weiss mentioned his name to the president of Rockefeller University, who invited Don to come to Rockefeller as a graduate student. Instead, Don went back to medical school, but that didn't last very long. He really liked research too much. The following winter, Ada Levy, who was fresh out of City College in New York and bound for Harvard University in the fall, arrived at Einstein to work as a lab technician. Don stayed on after work one day, ostensibly to help Ada clean up, though his real purpose was to ask her out for a date, according to Ada. So there we go. The following fall, Ada went on to Cambridge while Don started his research career as a graduate student at Rockefeller. Gerald Edelman was his Ph.D. advisor. A year later, the two married. They were not Gerald and Ada, Don and Ada. I just wanted to make that clear, yeah. They were determined to live together. Professional couples living apart was not exactly common in 1961. But a quaint university policy prevented married couples from entering Rockefeller's graduate program together. So Ada attended New York University instead. Can you imagine leaving Harvard to go to NYU? That was NYU's, they won and Harvard lost. But it was a way for them to be able to do the top priorities they had, which was to be together and to do science together. Both Olinses finished their PhDs and secured postdocs at Dartmouth Medical School. It's like a who's who of great universities. They had a son and later another, and we took the playpen to the lab in Hanover, Ada recalls. After the postdoc, they took jobs in the biology division of the Oak Ridge National Laboratory in Tennessee, where they were going to spend really the next 30 years together. Their research careers, they began their research careers and continued to investigating the protein structure of chromatin, the package that houses DNA in the nuclei of eukaryotic cells. And that's where much of their work was done. While at Oak Ridge, they obviously did sabbaticals both in London and also in Germany. But the key discovery was one day they were viewing a chromatin under a magnifying glass, and the Olinses saw what looked like beads on a string. Don called these repeating units the fundamental entities of chromatin, V-bodies, which later would be termed nucleosomes, which was really quite exciting. The other thing, there was a reference to their work, just to give you a sense of how significant their work has been. Roger Kornberg, who won the 2006 Nobel Prize in Chemistry, cited their work relating to his own work. So it gives you an idea of the importance of their work. It's interesting, I think Don will probably mention Ada's expertise as in microscopy. They actually have a collection of antique microscopes, which I didn't know, and I'm looking forward to seeing someday, which would be really terrific. they talked about their professional and their personal relationship as being one that they're okay to criticize each other because they learn from each other and don't take it too hard. After 30 years at ORNL, they left there. Don took retirement and moved to Maine to be closer to their children who had settled in Maine, and first worked at the Foundation for Blood Research in Scarborough, and then with Bowdoin College for a number of years. And subsequently, when that opportunity ended, they came to UNE, and we're really, really happy to have them here. So it gives me a great deal of pleasure to introduce two just exceptional people as well as exceptional sciences, Dr. Ada Olins and Dr. Don Olins.
Thank you very much, Bob. I appreciate that generous introduction. And I know there were people who in the audience who really enjoyed the playpen part of that because the person who was in that playpen is in the audience, so I'm really tickled about that. But I really want to thank the university and to thank you and our department for giving us the opportunity to continue to work. It's very special to be able to do the work that you're interested in, to have the freedom to explore whatever the work leads you to. And that's been a great strength for us and a great pleasure for us. So we've agreed as a part of this talk to also present some historical aspects. And I'll start with the micrograph that you see here. This is, of course, quite old, 1882. And I want to point out several things. Walter Flemming had a huge impact on our understanding of nuclei and chromatin. This is a micrograph, hand-drawn. There was no phone snap, snap, snap. It was very careful work, and I want to point out, I'm going to try to use my, my arrow on my computer so you can follow what I'm pointing to. This is a cell in mitosis, so there is no nucleus. The nuclear envelope breaks down during mitosis. But I want to point out the chromosomes here in orange, and the detail that Walter Flemming was able to depict, first of all, that there are two chromatids paired together, and then also these little particles in the substructure of the chromatin. And these are called chromomeres, and Don is going to show you some modern day micrographs, and maybe we still don't know exactly what a chromomere is, and some of our postulates based on our present day experiments. I also point out there's the spindle pole body, the microtubules going to the spindle pole body. The detail in this drawing is really fantastic. The other thing I want to point out is that two words in our daily usage on this subject, chromatin and mitosis, were coined by Walter Flemming. Chromatin meaning colored material, and mitosis is division of threads. So that's really a lovely, lovely word, and we use it every day. So I'll start with giving you an outline of the talk. I'm going to present the first two subjects, and Don will present the second two subjects, and give you a little game to play as we go through the talk, is to try to figure out what some connections are between the different parts of the talk. So first I'm going to talk about the discovery of the nucleosome. Dean McCarthy already has explained to you that that's the fundamental structure of chromatin. And then I will talk about nuclear shape. Don will talk about the surface of chromatin and some of the biochemistry of that. and our ideas about chromomeres, as I just mentioned. And then the last part of the talk will be about phase separation, which is a new concept, it's a very old concept actually, but it's a new way for us to look at the structure of chromatin and the material in the nucleus, a much more dynamic image, and not such a crystallized image. So without further, I'll just do a little bit of history, give you the first sighting of nucleus was by Robert Brown in 1831. So the history of chromatin is really not that old. Friedrich Miescher discovered nucleic acids, and you can see he had a little formula for it. It looks to me like a very small formula because we now know we have 2 yards of DNA in every nucleus in the cell. So think of DNA really as a very long, very thin thread. But this was the first step in identifying that material. Walter Flemming's I've already talked about. Kussel discovered histones, which are the basic proteins which bind to DNA, and I'll have a lot more to say about them. But it's interesting that even by 1941, the Cold Spring Harbor meeting on chromatin came to the conclusion that it was the histones that carry the genetic information, not the DNA. So the concept of DNA having our genes is really a fairly new concept. Hasn't always been there at any rate. It was in 1944 that Avery, McLeod, and McCarthy showed that it's actually the DNA that has the genetic information. So that was a very important milestone in the development of our understanding. And I just want to briefly mention Watson and Crick and Wilkins, who got the Nobel Prize for the DNA double helix for data that they stole from Rosalind Franklin. And but of course, the concept of the DNA helix and the way it can replicate is very fundamental to our understanding of the cell. In 1970-71, Don and I spent a year in London. At the time, we wanted to work with Pardon and Richards. They had just proposed a model for chromatin structure, and that model had the histones on the outside of a DNA helix. There was another model from Bram and Rees in the United States. Richards and Pardons were in London. But you can see that this is a structure which is very different from our present day understanding. One of the most important aspects is that we now know the DNA is on the outside of the histones and the histones are not coding the DNA. So with these models, we proceeded to try to learn more about chromatin structure. And I'd like to show you the microscope on which our first micrographs of nucleosomes were taken. This is an old Siemens microscope. And yes, every one of these knobs had to be adjusted in order to take a micrograph. And the lovely thing is, at the bottom here is a camera which held 12 glass plates on which you could take your images. And every time you finished taking 12 images, you had to open up, break the vacuum in the column, take out the film, and develop it in a darkroom and, in different solutions and so on. But you had to wait for the microscope to pump down again before you could continue working. It's a little different today from that. But I think Gary Larson really captured it. So I feel like that really depicts what my life was like in the early 70s. I'd go home and make dinner and come back to lab at night and work on the microscope. we had the great good fortune of working with Oscar Miller in Oak Ridge. He developed a method for looking at nuclear structures, which was very important in the development of our understanding. It involved swelling the isolated nuclei and centrifuging them onto a grid, onto a carbon film, and thereby spreading out the chromatin so it wasn't all condensed and bunched together so you could actually see it. So we did that, and we did something that we've done throughout our careers really, is to choose material that amplified something that we were interested in. And we used chicken erythrocyte nuclei where we knew all the chromatin was inactive. There was no transcription going, almost no transcription going on in that nucleus. So we knew everything was inactive. So that was the first thing that helped us to interpret it. And then I mentioned this detergent PhotoFlow, which was used to wash the photographic film after developing it, but Oscar Miller found that it really spreads the chromatin beautifully on carbon films. So those were the two things that really helped a lot. But you see, at this magnification, you can't say very much about the structure of the chromatin. And we would sit with magnifying glass on the prints and try to figure out what we saw. And one day, Don looked at these things. Gosh, look at that. It's all particles. So it was not helices, it was particles. So that was a very important realization. And here's a picture on a more modern electron microscope taken a few years later that shows you the structure of the DNA connecting nucleosomes. We called these structures new bodies at first, but nucleosomes was coined by Pierre Chambon and stuck as the name. Don at that time was teaching P-Chem and was very much involved with symmetry and crystallography and things like that. And he said, well, the DNA has a dyad axis, maybe the particles also have a dyad axis. And that turned out to be absolutely correct and was shown in crystallographic studies. So, well, let me put the crystal structure on here. In 1993, Jerry Bunick and Ed Uberbacher in Oak Ridge crystallized nucleosomes. The method that they used and the DNA that they used was usurped by Tim Richmond a little later, but I don't want to get into the politics of it. Let's look at the structure of the nucleosome, and I welcome you all to come up afterwards and hold a nucleosome. The symmetry is such, the DNA wraps around a core of histones. There are 8 histones in the core, and there's another histone, which I'll show you a little later, H1, which sits as the DNA exits. This shows the part of the nucleosome that is crystallizable. There are parts of the protein of the histone which are unstructured and don't show up in the crystal data. So this is an important part of chromatin structure. It makes it very dynamic. Those tails that come off, they're basic tails that come off the histones, and they carry the modifications that then fine-tune the activity in the nucleus. I just want to point out, here is the acidic patch, which is a region on the slide, the pink and yellow form an acidic patch. And that's a region of the nucleosome that a lot of proteins bind to. I've listed a few of them in this box here. We're particularly interested in it because we have an antibody which actually recognizes the acidic patch. And Don will mention that a little more detail. But it's interesting that there's a structure that attracts proteins and proteins can bind to and do in many cases. So here is the acidic patch on this side, and because of the symmetry, there's an acidic patch on this side as well. So the binding can occur on both surfaces of the nucleosome. Come up and hold a nucleosome afterwards. I have a cartoon here where the unstructured tails of the histones are depicted. And here H1 is not a core histone, but one that's probably involved more in higher order structure, but we don't really know yet how. And we don't really know how the dynamics of these tails affect the structure of chromatin, but clearly involved in nucleosome interaction. And I also want to point out that many of the epigenetic modifications take place on these tails. Okay, I'm going to switch subjects now completely and talk about nuclear shape. And some years ago, we decided to work on this, and partly inspired by this film, which is a neutrophil, a white blood cell. It's surrounded by red blood cells. And the neutrophil has a very important job to do. It protects us from bacterial infections. So you wonder what happens when a bacterium gets into our blood system. That's what happens. I'm going to run the movie a second time for you so you can, we really love this movie. It was made in 1950 on 16 millimeter film and fairly primitive microscope. But you can see what a neutrophil does. Now, the reason I'm showing you a neutrophil is that because this cell has to migrate through narrow spaces and in all kinds of ways, it's got a very unusual nucleus. Of course, it's the nucleus that attracted us to neutrophils. And let me show you a few images of human neutrophils and mouse neutrophils. And the thing to notice is that the nuclei are lobulated. They're not just a round ball. They have different portions, but it's one nucleus, even though it looks like little lobules. And in the mouse, it's even more unusual. It's an annulate-shaped nucleus. Now, one of the things neutrophils have to do is to leave the blood circulatory system and exit into wherever an infection might be taking place. So here's a beautiful electron micrograph taken by the Dvorak's of a cell, a neutrophil, exiting from the blood vessel through the epithelial layer of cells. So it's squeezing through a very narrow space to leave the blood. It's going after bacterium, just like in the movie, or something else that needs to be devoured. It's interesting that the nucleus the lobulated nucleus facilitates the squeezing through narrow spaces. And we've done some studies recently of changing pressure on the chromatin, and that seems to actually even affect the expression, what proteins are synthesized and what RNA is synthesized. So squeezing like that is also a very important function in neutrophils. But because we couldn't always get blood, we decided it would be best if we work with a tissue culture line. This is a cell line HL60S4, which we've been using for a good 20 years, I think. And what is so great about this cell line is that you can grow it in an incubator, in a petri dish, and always get the same kind of results. The control cells have big nuclei with prominent nucleoli. If you treat them with retinoic acid, they'll turn into granulocytes and they'll develop lobulated nuclei. If you treat them with 4-bol ester, they'll turn into macrophage. So we can control, to a certain extent, what the changes in nuclear structure are. And that's why we chose this cell, to study the changes, how the shape changes in a nucleus. So let me introduce you a little bit to what the nuclear envelope structure looks like. This is a cartoon. It shows the outer nuclear envelope with ribosomes attached to it. There are pores in the nuclear envelope. I just put a line there. It's, of course, a complicated structure which regulates flow in and out of the nucleus. But I want to show that there are many proteins in the inner nuclear membrane, and they form a layer between, they form a layer and then connect to another layer of lamins, which then connect to the chromatin structure. So all these are important players in determining the shape of the nucleus. I'm just going to concentrate on LBR. That's the protein that we've studied the most. It's a fascinating protein. It binds to lamin B1 and lamin B2. It also binds to heterochromatin protein alpha, heterochromatin protein 1 alpha, and it also binds to chromatin. So there are many interactions, and I'm not even going to mention the interesting interaction that it also is involved in cholesterol biosynthesis, but that's not going to be a part of anything I say. I didn't say that. Now, when there is a mutation in lamin B receptor, and this occurs as an anomaly in humans, and it's called Pelger-Hewitt anomaly. It's been known for quite a number of years. In the wild type, the neutrophil, as we've already seen, has a multi-lobed nucleus. The heterozygote has this interesting bilobed shape. And then the very rare homozygote has an ovoid nucleus. So you see, it manifests also in human patients and is an important understanding of nuclear structure. This is a cartoon drawn by Monica Zwerger to show the different parts of LBR. It's got 8 transmembrane domains. It passes through a lamina and then interacts with chromatin in various ways. When you knock down, and knocking down a protein is a modern way of learning how, what the functions of a protein are. You can manipulate the molecular structure of a nucleus and get rid of a specific protein. So we did an experiment knocking down lamin B receptor, and this diagram shows electron micrographs of the normal S4 line and the knockdown short hairpin line that does not have LBR in it. And you see, if we treat with retinoic acid, the normal cell gets lobulated nuclei, and if we do that with a hairpin knockdown and there's no LBR, the nucleus stays rounded. So this is one demonstration of the function of LBR in controlling nuclear shape. So just to summarize, I'd like to mention that when you do an RNA-seq and analyze the different RNAs that are synthesized, we find out that lamin B receptor is increased during differentiation to granulocytes, and lamins A, B1, and B2 decrease. And in the normal cell, nuclei lobulate when you go through the differentiation to granulocytes. I've shown you in a cartoon, and we've done a lot of experiments to show that the N-terminus binds to chromatin and the C-terminus is embedded in the lipid bilayer. And I showed you a little bit about genetic disease, although there are others that are interesting, especially in connection to the cholesterol biosynthesis. And with that, I'm going to build a bridge to the second half of the talk, a bridge of nucleosomes or maybe chromomeres. I'm not sure right now.
Well, first of all, thank you very much, Bob, for arranging this. And I know Karen is not here today, but she was really involved in this. And we appreciate what you said about us. Really, I didn't recognize that that was us that you were describing. It sounded like it was somebody else. Okay, so let me go. So what I want to do is talk about other features of chromatin, epichromatin, and chromomeres. But what I want to do is to continue on where Ada was. Ada left you believing that the surface of the nucleus, the nuclear envelope, is really a very important site. So on this next slide, what I want you to see are two micrographs. I have to get used to using this cursor. Where is it? Yeah, okay. On the right side is, this is actually a pretty old picture that we took quite a number of years ago, using a DNA-specific stain. So nothing else is being stained except DNA. And this is, again, the HL60S4 cell that is really our favorite. Oh, does the cursor disappear? Okay. Anyway, I want you to notice how smooth the surface is. This is DNA, only DNA that you're seeing. Notice how smooth the surface is and how there seems to be a line of DNA right at the edge of the nucleus. This is, you can't see the proteins. So the rest of the nuclear envelope would be here, and then the cytoplasm would be there, but they don't, they don't have DNA in them, so you don't see them. Then on the left side is a recent image that was collected by a collaborator of ours, Travis Gould at Bates College, who built his own super resolution fluorescence microscope, a STED microscope it's called. And this is the same cell type, years later, stained with a DAPI stain, which is a specific stain for DNA. And here, even at this light microscopic super resolution, you can see that the surface DNA really looks somewhat different. Okay, then Ada mentioned to you the antibodies that we have been using. We collaborated with a person, Mark Monastir at Temple University, who was interested in the mechanisms of autoimmunity and had a strain of autoimmune mice, and made a lot of different antibodies from this strain. And gave them to us, and we tested them on in this case, it's a different cell line, U2OS, which grows attached all the time, whereas the HL60 cells are in suspension. But I'm not going to distinguish between them. I mean, U2OS cells cannot be differentiated, whereas HL60 cells can. Okay, so this is the cell cycle. And this is using a method. I don't know how many of you know the method. It's a very old method, actually. It's called immunofluorescence microscopy. You basically make sure that your antibody is directly fluorescent by putting a dye on it, or indirectly fluorescent by using a second antibody that reacts with the first antibody and has a fluorescent dye on it. And then you look at the cells under the microscope, and you have to use fixed cells and permeabilize and react it with antibody. There's a whole rigmarole you have to go through, it's a beautiful way to see where antibodies are localized. So the red color is the antibody localization. The name of the antibody, beautiful name, PL2-6. I actually don't know where that came from, but it stains the rim of the chromatin. We know that this is chromatin staining. I'll show you later, because we know it from the essentially the tomography, crystallography data. We have other data that it's really the chromatin that's reacting with it. So here you go through the cell cycle, and you can see how nice and sharp this edge of chromatin is surrounding the blue, which is the rest of the nuclear DNA. It's a DAPI, which stains DNA blue. Okay, then we go through the cell cycle. You get the beginning of prophase. The nuclear envelope is beginning to break down, and you can see these where it's not as smooth as it was. Now you're into metaphase, mitosis, and there is no nuclear envelope anymore, but for some reason, the outer edge of this cluster of mitotic chromosomes is stained, goes through the cell cycle, then finally the daughter nuclei separate from one another, and this is now at late telophase, when there's new nuclear envelopes that you can't see. You're not looking at nuclear envelopes here, you're only looking at the surface of chromatin. Okay. Now, that's what you see when you use a bivalent antibody called PO2-6. Now, so this is a sort of a scheme of what a bivalent antibody looks like. It's got a light chain, the dark blue, 2 light chains, and 2 heavy chains, the lighter blue. And the binding site of each of the antibodies at the end of these, what they're called is FAB fragments. So it's a bivalent antibody. And that's what you saw in the previous slide. However, what we wanted to do, for a number of reasons and a lot of biochemical studies that we did that we're not going to mention, we decided what happens if you make a monovalent antibody from this antibody, and you use an enzyme called papain, which is a protease, and clips it, and you can isolate this monovalent fragment. So it's only got 1 binding site, not 2. Okay, and now what do you see? Now what you see, well, the top row is just the bivalent antibody, PL2-6. You can see it's staining the periphery like I showed you before. Here's the DNA in blue, and there's another antibody that we've introduced in this experiment. That's a H1 antibody. I just showed you how H1 binds to where pretty much the DNA comes in. And this is one. There are several H1 antibodies in these cells. This is H1.5. That stains differently. It shows a punctate structure. This is the interphase nucleus. Okay, the same one, and you can see then this is a merge of the different colors. Okay, but again, the PL2-6, the bivalent antibody, is on the outside. Now, if you look at the FAB fragment, you get a totally different picture in the interphase nucleus. Now you're getting staining throughout the nucleus in these punctate matter, just like H1.5. Here's the merge image. If you look at a mitotic chromosome cluster, you don't just see the outside of this cluster stained. You see lots of dots all over along the chromosomes. This is one set of chromosomes. These are just sequential slices, optical computed slices. This is all done in modern microscopy, which is digital. You calculate the 3D structure, and then you view single slices. So we, because we love history, we said, okay, we got to give a name to this. It looks to us an awful lot like what Walter Flemming saw. Let's just call it chromomeres, give it a modern context. And so that's what epichromatin is, the name we gave for what the bivalent antibody sees. Epichromatin, surface like epidermis, the skin. So it's the surface chromatin. And then the chromomeres are these little particles. Okay, so now the structure takes us way into modern day structure, and how we know about this. Well, this structure was actually determined by X-ray crystallography, but how do we know where the FAB binds? That you can see here. This is an incredible method where the complexes are frozen in ice, and you collect thousands of images, and the computer sorts it out, and you can get actually atomic level resolution. It's called cryo-EM tomography, and there's a beautiful study that's been done. You're looking here, you're looking down on an FAB sitting right on the face. Here you're looking at two of them binding to the acidic patches here, and different views of it. And here is a model of how the bivalent antibody might be binding to the to a dinucleosome. Okay, so what you've got in this movie is you've got two nucleosomes connected by this region of DNA, which where it's thought parts of H1 binds to it. So this is the two nucleosomes, and this red sprinkling here is the acidic patch. And so now you can see the bivalent antibody binding to it. Okay. Oh yeah, let's get out of here. So finally, summary of this part. And what I want you to remember is the epichromatin is stained by the bivalent antibody. The chromomeres are stained by monovalent antibodies, and the difference between the staining is probably tied up to the exposure and the geometry of the nucleosome acidic patches. Okay, well, I jumped to the next slide. Let me just go back, see if there was something else. No, so the exposure and proper geometry. Now, the last part of our talk takes us back again into the pages of history. So, phase separation of chromatin, as Ada pointed out, is a very hot area. I should also, as a sort of a joke, I should say that cryo-electron microscopy tomography is also a very hot area. Okay, but this is another very hot area in the field of modern chromatin research, nuclear research, and cellular research. Phase separation. Otto Bucheli in 1892 proposed that protoplasm is really in emulsion. When he looked down these microscopes, the late 19th century microscopes, which were beautiful German microscopes and had wonderful numerical apertures, and he looked at living cells, he could see that it looked like an emulsion. There were little granules and vacuoles, and they were moving around. So his theory was that protoplasm is an emulsion. Now, an emulsion, what is an emulsion? Well, I guess the best way to describe it is to say you've all had vinaigrette salad dressing, oil and vinegar and spices, and you give it a good shake long enough, and you get this suspension of tiny, tiny particles. Well, that became the idea, really, for what protoplasm is like and what nuclei and what structures are like in cells. So now I'm going to show you a story that has been evolving in our lab over the last few years. Basically, what we do is we use this immunostaining method. And you'll recognize PL2-6 and DAPI. All right, so on the left side of this is just the ordinary cells, not exposed to anything. They were in medium and fixed and put down on a slide. And now they're stained with the PL2-6, which stains only on the outside of the chromatin in the interphase cells and the outside of the cluster of chromosomes in mitotic cells. And the DAPI stain is sort of bluish or cyan. Now, if you put cells in sucrose, now why did we use sucrose? Well, sucrose does not get into cells, mammalian cells. Rather, it has a high osmotic pressure and it draws the water out of the cell, and the cell is becoming dehydrated. This had been an issue that people had been studying for a number of years, mainly focusing on what it does to the interphase nuclei. In fact, nobody had looked, to the best of our knowledge, at mitotic chromosomes. And here you can see this is the hazy DAPI staining. I'll show you a higher resolution picture in a minute. The hazy DAPI staining in interphase nucleus and the mitotic chromosomes. And high sucrose, high osmotic sucrose, the chromosomes collapse. And we call this congelation, from the word congeal, but we like it because the word gel is right in the middle of it. Whoops, I gotta go back. Excuse me, so here in 0 millimolar sucrose, you can see mitotic chromosomes, the DAPI, and you can see the PL2-6 staining on the outside. This is a hazy sort of interphase nucleus. And in 300 millimolar sucrose, you have this gelatinous-looking structure surrounded with the exposed epichromatin. Okay, another example, a beautiful case, is the protein Ki67, which is a mitotic marker. It only shows up mitotic chromosomes. It does also react with nucleoli, but I won't have time to mention that. So these are three sets, clusters of mitotic chromosomes in 0 millimolar sucrose, just regular tissue culture medium, fixed in that. And now the DAPI is blue. We had to change the color back and forth. This is all false color anyway, because we now have 3 dyes here, and we have to see all 3 of them. So anyway, what you can see here is the PL2-6 staining on the periphery. You can see the Ki67 in green in between the chromosomes. You can see it down here at a higher magnification. And then in 300 millimolar sucrose, when the mitotic chromosomes cluster, they collapse, they congeal. You can see it here at the bottom. Here, the boundary of the DNA of the chromatin, and on the outside now is the Ki67. So it's moved from the inside to the outside. This is phase separation. Okay. And here's another example, histone H1.5 in the absence of sucrose. Since it's got, you can see the DAPI here. So it's a little bit harder maybe to see the chromomeric structure of the mitotic chromosomes. The polka dots on the surface, the epichromatin on the surface of the cluster. Now in 300 millimolar sucrose, you can see the histone H1 has moved to the outside. It has phase separated. Okay, and a little bit more. It isn't only sucrose that does it. other molecules, other chemicals that don't get into cells but dehydrate the cell, like sodium chloride, will dehydrate the cell and will have the same effect. And 150 millimolar sodium chloride, since it's got a sodium and a chloride, and we won't get into the chemistry of colligative properties, that's about comparable to 300 millimolar sucrose, and it has the same dehydrating effect on the cells. Okay, so now my summary. Let's see how we're doing. Okay, we're near crashing into the end. So, hyperosmotic stress with sucrose or sodium chloride dehydrates the live cell. And what's the mechanism of this segregation and collapse? We think it's changes in the molecular bonding strength, and also certain proteins dissociate. This is an issue that we would be happy to discuss at the end of the lecture. Okay, the cells are still alive. We've done viability measurements on the cells even up to 3 hours in sucrose, and they're still alive. So now what we want to find out is, can we examine the reversibility? So while we were preparing this lecture, Ada came up to me and said, look what I found. And it was a page from a magazine she was looking at about cooking. It was a cooking magazine. So I want to show you that. This is an important part of our lecture. Okay, well, oops, I forgot it again. So here we have the title, 1 tablespoon of molasses is the secret that made our make-ahead vinaigrette unlike any other. So this is the part I like, end separation anxiety. So if you add molasses and shake it up, it will remain as a suspension. It won't have phase separation. So we're not thinking of adding molasses to cells, but this is an indication that there are ways to stabilize and destabilize structures in the cell to make them phase separate, as happens with the nucleolus is actually normally phase separated. And finally, I know we've covered a lot of things and we haven't covered them completely or always clearly. I want you to know how we feel about what it's like to do research on the nucleus. You can see the nucleus is an incredible structure. And really, it depends on the technique that the scientist is using. They might say it's like a wall, or it's like a snake. So it depends a lot on the methods that we have employed. Finally, I want to give credit to the many, many people who we've collaborated with, who have helped us on these projects and years. You can see scattered around the world, and in fact, we have been told some of them are watching this right now. And most of all, we want to thank the College of Pharmacy and UNE, Dean McCarthy, Provost House Connect, and our chairperson, Olgin Gavench, and our colleagues in our department for really making us feel very comfortable and able to continue searching. Now, I hope I've left enough time, probably not, and that you folks can feel free to ask questions to us here or afterwards, and can look at what a nucleosome looks like. And thank you very much for listening to our talk, and to any friends who are watching from afar, we give our best wishes. Bye.
I think we have time for a question or two. Folks have? Yeah, please.
That was an amazing lecture. Thank you for that. So I actually had two kind of interrelated questions. So I'm assuming, based on the cell type and cell function, you see differences in the chromatin formation to the outside. So my first question is, if you are looking at something like a cancer cell or a senescent cell, are there patterns that you see? And are they catalogable to distinguish those kinds of cells within a tissue? And my second question was, on that very last part where you showed the congealing, it looked like the euchromatin, the H1, was to the outside, and the heterochromatin was to the inside. So based on again, similar questions, based on the cell type, and you said the cell stayed alive, so those parts, if the cell had been a liver cell to begin with, or a neuron to begin with, the heterochromatin that had been in that original cell, that's what goes inside, or does that change? And I guess you can tell that by the gene expression.
Wow. I'll take the first crack, and then Ada will tell you that I was completely wrong. Let's say, in fact, we're not seeing the heterochromatin move away. I think you must have been talking about the histone H1.5. No, I don't know what else that H1.5 is bound to, but then there's no DNA out there. That's outside the DNA. So it's histone H1.5 may be bound to something else, but the heterochromatin, if that still exists in the, or how it exists, would be still, in our view, within the boundary that's defined by the epichromatin. So I don't think I'm not saying there haven't been changes in the heterochromatin. And in fact, these are the kinds of experiments we're looking at right now. We're using another method called Hi-C, which was developed it's called an all-against-all method. You ask the question, where do all sequences of DNA bind to with respect to all sequences of DNA? And it's a very good method for defining chromatin compartments. And we are still trying to understand those experiments. That was the second question.
One of the things we're trying to do is to analyze the histone modifications and see what the changes are when we put the cells in sucrose. Is this working?
Yeah.
So that data hasn't been completely analyzed yet, but that's another way to access the question of heterochromatin versus the euchromatin. I think we see indications that there's still replication going on in the congealed cells, or nuclei. So that's something we've seen, which is surprising and interesting, and I think the fact that the cells are still viable in viability tests in the sucrose is also a sign that things aren't totally disrupted. I mean, when you see all the H1 leave the chromatin and be outside the chromatin mass, then you begin to worry that this is never going to be a reversible state, but it seems like it might be.
Can I come back to the first question that you were asking? I think in a way you were asking, can you tell the differences between the chromatin in different cells, and maybe differentiated cells versus undifferentiated cells, or different cancers, or neuron versus liver cell or something? The answer is there are tremendous differences and there are big similarities. It's really, the nucleus really is an elephant. There are so many other features you have to consider. There's the epigenetics, that's the histone modifications. And also the DNA epigenetics, those are the DNA modifications, methylation. There are the histone variants, and there are all the other non-histone proteins that may or may, and so it's incredible. It's incredibly complicated. And so, yes, there are general patterns. And I'm going to, if I have the time, I want to show you one more slide. Yeah.
About using the PL2-6 as a probe for the structure of chromatin, and that is that we see peripheral staining like that in all kinds of cells, in plants, in fact, even in plant cells, in yeast, in what are some of the other cells we've done? But it always stains the periphery. Yeah. So there's something about, maybe it's a kind of surface tension on the surface of chromatin that orients the nucleosomes in such a way that the acidic patches are all pointing out in some ways. So there are some common elements, I think, in nuclei.
For many, many years, in a way, it was, well, we felt that the nucleosome, in a way, was the end of highly ordered structure in a nucleus. And beyond that, higher levels, and to a large extent, this is probably still true, chromatin has such a plethora of conformational states. The question is, can we look for any other repeatable pattern, structural pattern, that could be common between various different kinds of cells? And we think the chromomere is, and we didn't discover the chromomere, and we're not the only ones who we gave it a name, which some people may not like, I don't know. But the point is that from this method high C, it is clear that there are points on chromosomes where interactions are focused onto themselves. They're very narcissistic at that place. And then there's a space, and then there's another cluster. And we might call those, they've been referred to as TADs and compact domains. Various labs have different names for them. And they do seem to have some patterns, even between different cell types, some similarities. So there does seem to be another level of packaging there, which is, now, years ago, scientists discovered something called lampbrush chromosomes. And lampbrush chromosomes are chromosomes seen during meiosis, when a lot of RNA is being made and big loops come off of chromosomes, and they come off at discrete points. And those probably are similar things, chromomeres, TADs, whatever you want to call them. So that's probably an important next level of structure. And then there are the chromosomes, the interphase chromosome territories that were described by the Kremers not too many years ago, where a chromosome has its own integrity and individuality. So all of these things enter into the there are similarities between cells and differences between cells. Can you tell a cancer cell from a normal cell on the basis of this? No, I don't think so. Maybe epigenetic modifications will help, but it's still thought to be mostly genomic changes rather than epigenomic. But there are some epigenetic cancers that have been described. Those were big questions.
All right. As we close, I just wanted a couple of things. Number one, I want to thank Danny Houston for all her assistance today. Thank you very much for putting this all together.
Yeah.
And one thing I left out of my introduction that I wanted to say for the end was that Don and Ada will be being nominated for the Vohweiler Research Award, which is a Lifetime Achievement Award given by the American Association of Colleges of Pharmacy. To my knowledge, and I've been in pharmacy education for 33 years, no research team has ever been, this is an award given to one person, but we're nominating the two of them for the first time in the awards history. So no guarantees I'll receive it, but boy, it's a pretty powerful package, as you can see. Please join me in once again thanking Don and Ada for a wonderful presentation. Thank you.
Thank you very much. Thank you. Thank you.